All right, insects are amazing animals, at least I like them. A lot of them have pollination and do other things that are useful for humans, but there's also a lot of very destructive species, such as the New World screwworm up there. Now, this once terrorized the cattle industry because they lay their eggs along runes on cattle and sometimes humans, and their larvae feed on living flesh. So this species was actually completely eradicated from North America using sterile insect technique, whereby males are reared up in huge numbers, irradiated to make them sterile, released into the wild, they mate with wild females, they don't produce any viable offspring, and then the whole population crashes. So this species is now gone from the US. Now this technique doesn't work as well for other insects such as mosquitoes. So new genetic methods are being developed to do a similar thing. And I'm using the model system Drosophila, the fruit fly, to study a transgenic system to make males sterile. So this can be controlled using the antibiotic tetracycline, which prevents the expression of a lethal gene that's been inserted into these flies. So with tetracycline, we can rear up these flies in huge numbers in the lab. Without tetracycline, this lethal gene is expressed and gets passed on to the next generation, killing any offspring. So this works really well in the lab in limited lab strains, but it hasn't really been tested against a wide genetic background. So for my research, I've been taking different genetic backgrounds of this fly representing the global diversity of this species and testing to see its effectiveness. So these bars each represent a different fly line, and you'll see that overall it's fairly effective. We get generally less than 1% surviving, but we do get some survivors. And there's some lines where nothing survives at all. So it's very effective against those genetic backgrounds. There are survivors. These survivors can continue to produce offspring. So we need to figure out what's going on with those. So the next step of the project is to determine what genetic backgrounds contribute to that survivorship and how we can mitigate that in the future. Thank you.